All right, so we had to stop unexpectedly there for a second. I apologize for that very short section on replications. We're going to pick up here where I left off a moment ago. And we were talking about the overall accuracy of the DNA replication mechanism and the fact that the cells have multiple ways to fix DNA errors or mistakes or mutations when they do occur. The overall background rate of mutation was around once out of every 10 million or so attempts to copy DNA. And then when mistakes do happen, they'll be fixed most of the time. Now, mutations that don't get fixed are the ones that we talk about actually having an effect on the phenotype or physical appearance or behavior of the organism. Mutations can be relatively minor and have really no effect. They can be sometimes lethal, where they have a huge negative impact. Sometimes they can have a not quite lethal effect, but also have a bit of a negative effect as well. It's somewhat more minor. And sometimes mutations can be beneficial, although those are, by far the minority of mutations are the ones that are beneficial. What we're going to talk about for these next few slides here are the different kinds of mutations that can occur. The first type of mutation is called a point mutation, also usually synonymously used with the term substitution mutation, where we get the wrong base plugged in at a given point on the DNA chain. Now, because we haven't really covered how transcription and translation work, you're going to kind of have to take my word for this for right now. But suffice it to say that sometimes if you switch in the incorrect base on a new DNA strand, you might actually end up not changing the phenotype that much. In other words, you might actually get the same protein you should have gotten without the mistake, even though you actually had a mutation in the DNA. And so again, with the details of that, the ins and outs, the intricacies of this, we're going to get to that here in a couple of chapters. But for right now, just know that the genetic code, in terms of how we go from DNA to protein, is somewhat redundant, and we can actually change the DNA without having a big change on the overall structure or function of the protein that it's supposed to produce. Right? So point mutations sometimes can actually be completely neutral and have no effect at all, and other times they might end up producing a protein which is somehow different, whether it's beneficial or harmful or lethal, depends on the specific details of that mutation. Now, in insertion mutation, and it's kind of like close cousin here, the deletion mutation, these two types of changes that occur are typically pretty catastrophic, because what happens here, if you add in a base or 2 or 4 or 5 or 7 or 8, or take out any of the same numbers, that will greatly change everything that occurs from that point on throughout the entire rest of the protein. And the reason for this is that the genetic code works in multiples of 3. So if you were to add in 3 bases, or 6 bases, or 9 bases, or 60 bases, or take out the same numbers, yeah, you'd be adding in or taking out individual amino acids, but otherwise the overall sequence of amino acids in the protein would be completely normal. On the other hand, if you take out just one base, then all of a sudden the 3-letter sequences that occur after that point are completely screwed up. And that's going to have a very, very negative impact on the overall protein that's produced from that. So deletion mutations, insertion mutations tend to be pretty catastrophic unless you happen to just insert 3 or 6 or 9, or take out 3 or 6 or 9, or any other multiple of 3, then the mutations, potentially at least, don't necessarily have to be quite so severe. Now, an inversion mutation occurs when a piece of DNA is removed from the original strand, the original chromosome, it's flipped around like 180 degrees, and it's put back in place. Now, you might expect that to be, again, pretty disastrous for the overall effect on the phenotype, but it actually doesn't necessarily always have to have that big of an impact, because if you do this to the entire gene, if the entire gene is removed from the chromosome, flipped around, and stuck back in, the sequence of letters is still the same. It's just reading in the other direction now to get that sequence. And that's perfectly fine. You can read DNA left to right or right to left as long as the order of letters is relatively the same, right to left or left to right. It doesn't really matter how the gene is oriented in the chromosome. On the other hand, if you just take out half of a gene, flip that around and stick it back in, now the gene's letters are in a completely different sequence. Right to left or right left to right, it doesn't matter. They're in the wrong sequence. Therefore, the protein is going to be greatly affected in that case. So the main thing is here, if you're doing an inversion mutation, are you doing the entire gene, or is it just part of the gene? If it's the entire gene, it's not necessarily going to have any effect at all, versus if it's just part of the gene, then you potentially could have something pretty severe in terms of a mutation there. The final type of mutation is a translocation mutation. Translocation mutations occur where a gene is removed from one chromosome and transplanted onto another chromosome somewhere else in the genome. All right, so translocations, again, like the inversion mutations, don't necessarily always have to be that significant. If you move the entire gene from chromosome 9 to chromosome number 17, it's not a big deal. But if you only move half of the gene from chromosome 9 to chromosome 17, well, now you're going to have a problem because now the gene is effectively broken. It's been split in half, and now it's not going to create a correct protein. So again, it depends on the extent of the mutation as far as how severe or not that mutation might be. Excision repair is essentially referring to generalized DNA repair, where it's really just like proofreading anything that you've ever written, right? You look for mistakes that are made, you find them, you take out the damaged part, and then you replace it with the information that's supposed to be there. And that's exactly what happens with generalized excision repair in DNA, is that mutations or mistakes that were made are removed, and they're replaced with the correct sequence that's supposed to be there, thereby reducing the effect of the mutation or eliminating it completely. All right, so that is really about all we're going to say in this chapter. From here, we're going to move on to talk about cell division, which is both mitosis and eventually meiosis. So that will be kind of our next series of topics. But for now, we're going to wrap up on DNA structure and function and chromosome structure as well, and we'll move on from there.